Good morning, everyone, and welcome back to the cycle of conferences of the Costa Rican Center for Continuing Education in Clinical Microbiology. My name is Andre Montero, and I'm blessed to introduce the lecture for today. And after you have in the last two lectures about lentiviral vectors with Dr. Christian Smerlo of University of Navarra and Dr. Marco Ruela for University of Pennsylvania. We are now moving on to the CAR-T cells field, and we really have here an expert on CAR-T cells who's giving the talk today. He's a professor and investigator of gene therapy at the Center for Transfusion Medicine and Gene Therapy in the University of Freiburg. As you know, CAR-T cells is a very interesting area with a promising future around the world. And CAR-T cells are genetically engineered recombinant T cells that are being used with a great degree of success in the treatment of some types of cancer. It's a pleasure to introduce to Dr. Jamal Al Zu Bi. He's the team leader of the Immune Cell Therapy Department and has several publications related to CAR-T generation tools to improve performance of different therapies. And his science is dedicated on target cancer therapies and how to translate them mainly to the clinical use. So Jamal, thanks for joining us today, and the floor is yours.
Thank you very much, and good morning, everyone. From my side, thanks, Andrea, for this kind introduction. And the title of my talk today is about manufacturing CAR-T cells for clinical use, challenges and opportunities. So just a general introduction for the concept and the different generation of CAR-T cells and CAR immune cell therapy. So chimeric antigen receptor composed of a single chain fragment variable here consists of an antigen binding domain, antigen recognition domain, and this antigen recognition domain as a form of single chain fragment variable, SCFV, if used to CD3 zeta domain in the first generation CAR. In the second generation CAR, this concept allowed to introduce one co-stimulatory domain, mainly CD28 or Form1BB co-stimulatory domains, among others. In the third generation CAR, these two common co-stimulatory domains were fused together with the CD3 zeta. However, the most commonly used CAR-T cells in the clinic is based on this second generation CAR construct. And of course, the advantage of such a concept is the clinical efficacy that we have witnessed for treatment of hematological malignancy particularly. However, several limitations came alongside of this therapy, such as low CAR-T cell persistence, especially for solid tumor, the presence of endogenous T cell receptor, and this allowed the development of off-the-shelf product. Of course, the purity and the quantity of the generated product is a major concern in the field. Today in my talk, I will touch these limitations and show you how we can modulate and target solid tumor in one hand, and of course, how we could supply a high purity and quality of this CAR-T cell product as off-the-shelf ready-to-use products. So manufacturing CAR-T cells particularly mainly started with autologous sources, meaning that the patient will subjected to apheresis, and then T cells will be isolated, activated, and then transfused with the lentiviral vector or a gamma retroviral vector encoding for the CAR of interest. And then these CAR-T cells expanded, and then after several QCs to check the performance of these CAR-T cells, especially in vitro or in vivo, these CAR-T cells can be infused back to the patient. However, from the manufacturing perspective, this process is rather complex. It has several open steps and risks for contamination, not always possible to have a GMP grade materials, and of course, cost and manufacturing failure happened in these clinical trials. From the biological side, there are variation in the quality and the quantity from patient to patient of the starting T cell material. And as an example, 9% of the patients eligible for chimera therapy did not receive their therapy just because the quality or the quantity of T cells to start the transduction with was very low. And of course, there are an example for B cell contamination in the product. And this is here, for example, the introduction of the CAR to a single leukemic B cells, CD19 targeting CAR. It happened that this scenario led to the masking of the CD19 antigen and persistence of this single leukemic B cells, and the patient died from this therapy. So, however, we believe that it's important to unify the T cell source and, of course, start with the healthy allogenic donors to have a robust manufacturing, and this will allow us to stock the CAR-T cell product and be available right away as off-the-shelf product. However, there is still a need to eliminate the alloreactivity, and to achieve this, I think several concepts developed in the past to target the TCR alpha locus, the track locus, using several nucleases such as zinc finger nuclease, megatalin or talin nuclease, CRISPR-Cas9 as well, and many other colleagues among us as well try to combine this track targeting to provide off-the-shelf product together with other hits such as targeting CD52 or targeting LAG3 or BD1 to generate non-alloreactive and enhanced CAR-T cell therapy. However, as I highlighted, this strategy also mediated not only for the knockout of the track locus, but also with the knock-in strategy to have a physiological CAR expression. And it's important for us to know which nuclease we have to use in order to achieve this off-the-shelf CAR-T cell product. For that, we have developed an automated process to generate universal CAR-T cells, and we have used the Clinimax Prodigy. This is a device available from Meltini with an electroporation unit here. And this protocol looks like this, where we select T cells here at day 1, and then activating these T cells from healthy individual, and then transfuse them with an antiviral coding for the CD19 CAR at day 2, and then at day 4 we deliver the TALEN mRNA to the T cells, to the CAR T cells here through this electroporation unit. And then after that, we expanded the product in the Prodigy device for 14 days. And then we applied one step to eliminate TCR alpha beta, called TCR alpha beta depletion. And then we characterize our product in terms of phenotyping and functionality. And again, as I mentioned before, so which nuclease we have to use to target the track locus to eliminate the T cell receptor at the T cell surface. And for that, we have looked first into the activity and the specificity of the TALEN nuclease versus the CRISPR-Cas9 nuclease. And as you can appreciate here, for example, looking into the knockout efficiency, CRISPR-Cas9-3, guide RNA-3 complex with the Cas9 protein led to around 74% knockout efficiency, comparable to the TALEN mRNA that we have electroporated. CRISPR-Cas2 led to lower knockout efficiency. That's why further we looked into the specificity comparing the TALEN nuclease versus the CRISPR-Cas9-3 in this case. And when we looked into the specificity, we looked into amplicon sequencing for the predicted off-target size, up to 20 off-target size. And as you can see here, the TALEN mRNA did not produce any off-targets for the predicted size. However, the CRISPR-Cas9 guide 3 led to 3 statistically significant off-target size. That's why in this study, we focused on the TALEN mRNA to knock out the TCR locus. And of course, there was a need to evaluate how we could deliver these mRNA into T cells. And to do so, we have different settings of electroporation that we would like to test. And that was done in a small scale experiments. meaning that we electroporate the T cells either with the setting number 1 or 2 and 3, and we looked into the cell number as a readout for cell recovery after nucleofusion. And as you can see, up to 12 days, setting number 2 or 3 led to that the T cells recovered after the tailin mRNA transfer over time, but not, for example, setting 1. That's why in all subsequent experiments, also on the larger scale, we use setting 3 to electroporate the T cells. Of course, we looked into the percentage of CD3 negative cells, in this case as a readout for the TCR knockout, and we did not observe any differences of whatever setting we used. All of these settings led to high and efficient knockout efficiency at the phenotype level, where we achieved above 80% of the cells were CD3 negative. Then we moved one step ahead and we tried to adapt the full-scale protocol and large-scale protocol in the prodigy itself. And as you can see here, a pre-depletion, we could achieve around 42% knockout efficiency as evaluated by looking into the TCR alpha beta expression here in this case. However, post-depletion, we were able to achieve 100% TCR alpha beta negative T cells in this case. And this is the quantification of the several runs we did in the prodigy device. As you can see, we evaluated further the enders, meaning that insertions and deletions at the off-target site, where it matches the phenotype that we observe. After depletion, we were able to achieve around 100% at the gene level, where we knocked out this specific target. Furthermore, we looked into the functionality of these TCR-free CAR-T cells and compared them in this setting with the transduction-only condition, meaning T cells transfuse only with the CD19 lentiCAR, or pre-depletion, meaning a mixed population of CD19 CAR positive, TCR positive or negative T cells. And at the third condition, which is our condition of interest in green here, you can see post-depletion, where all of these CAR-T cells were depleted for the TCR alpha beta expression. And we have used here as an example a cell line expressing the CD19 positive antigen, and we looked into the cytotoxicity, where we could achieve similar levels of elimination for the target, whatever we used, either the transduction only, similar to the pre-depletion, or the post-depletion CAR-T cell, TCR-free T cells, at different ratio, either 1:1 or 1:5. In this case also, when we culture them with the K562, which is a cell line lacking the antigen, we did not observe any cytotoxicity, indicating again the high specificity of the CD19 CAR targeting CD19 positive targets. We have also looked into the cytokine release from these supernatants after the co-culture happened, and you can see similar levels of cytokines released from all of these products here, especially the TCR alpha beta depleted cells in green. And we did not observe any cytokine released with an antigen negative cell line in this case, either for interferon gamma, IL-2 or GM-CSF. This indicates that the tailor-edited TCR alpha beta free CAR-T cells perform equally well as non-edited CAR-T cells in cytotoxicity and cytokine release assays. In summary, for this part, we were able to establish an automated GMP-compliant manufacturing process to develop gene-edited CAR-T cells using the Clinimax Prodigy fully automated system. And we were able to disrupt the track locus to provide off-the-shelf CAR-T cell products that can be used as a bridging therapy, for example. And the final product was safe and based on at least the rough look into the specificity profile of these tailin, and was effective in terms of tumor killing cytokine and T cell phenotyping. Due to time, I didn't show you all the data, but I can confirm that our final product were pure in terms of TCR alpha beta expression. And of course, in terms of innate cells in the final product, where it's below 0.1%. Now I switch gear to talk about the solid tumor, and here you can see the scheme again for the treatment of leukemia using CAR-T cells, where these CAR-T cells can be leukapheresed and then transfused with the the T cells can be leukapheresed and then transfused with the CD19 CAR. And then this setting here in several clinical trials led to elimination and complete remission and partial remission when targeting, for example, CD19 positive tumors. And some responses also have observed when targeting several leukemic antigens, B-cell antigens expressing either CD20, CD30, and so on. And this indicates the success of using these CAR-T cells to treat hematological malignancies. But when we move to the solid tumor, the picture is a bit disappointing, and only partial responses were observed. And of course, subjective responses were observed, especially to treat prostate cancer, as you can see here. 5 treated with the PSMA CAR T cells, and then only partial responses among this small cohort of patients were observed. And we focus on our lab to target the prostate cancer, and this is a bit hard to treat a tumor. And of course, the relevance for such a target and the prostate cancer is that more than 1 million new cases per year diagnosed and above 300,000 deaths per year observed, and it is the 5th leading cause of cancer death. And of course, our target of interest is the prostate-specific membrane antigen. It is expressed in all tumor stages and overexpressed in advanced and metastasis disease. It has a low expression levels profile in the brain or salivary glands and expressed in the endothelium of other malignant tumors, which make this target really ideal to treat prostate cancer. So what we did in this regard, we teamed up with our colleague in the urology department here in Freiburg, Philip Wolf, who developed anti-BSMA monoclonal antibodies. And he showed that the antibodies he developed were has a high affinity to the target and high specificity to the BSMA target. And what we could do is to have these antibodies and clone them in a 2nd generation CAR. And we have characterized in this regard 2 CARs, one with the CD28 so co-stimulatory domain and one with a 4-1BB co-stimulatory domain fused to the CD3 zeta, of course. And this work was published here. And so I just go through what we have, the main finding we found for these novel CAR-T cells targeting PSMA antigen. And the first step, we of course want to see how far we are from the benchmark CAR, which is known here as a J591. This is a non-CAR with a single chain fragment targeting PSMA as well, and used in the clinic. And the efficacy of this CAR is not, it's the state of the art in this case, also for using in the clinical trials. And for that, we have used here our CAR, here termed as a D7, and here you can find CD28 co-stimulatory domain and CD3 zeta, 2nd generation CAR, and we compare it also to the J591 CAR construct, similar to our construct, except that the single chain fragment derived from the known J591 single chain fragment. We have activated the T cells, transfused them at day 3 with the gamma retroviral in this case, encoding each of the CAR, expanded these cells and characterized them after day 9. The first step, we did a cytotox assay, meaning that we cultured the target cells expressing PSMA antigen with either the D7CD28 CAR, called here CAR-28D7. This is our CAR, and this is the state-of-the-art CAR here, J591 in green. And as you can see, we mediated higher cytotoxic potential of these CAR-T cells in eliminating the target cells at different effector to target ratio. However, when we cultured them at 2-to-1 ratio of effector to target, when either our CAR or the state-of-the-art CAR showed similar killing up to 100% for the target. And of course, this combined with the higher levels of cytokine release, such as interferon gamma mediated for our CAR in red here, as compared to the J591. From these experiments and others as well, we concluded that our D7-based CAR is more active than the clinically used J591-based CAR T cells, at least in vitro in this case. Then we moved one step ahead and changed the transmembrane, the co-stimulatory domain in this case, either with the 28 or with the 41BB, and compare the functionality of these two different CARs based on the D7 single chain fragment, our CAR, in terms of cytotoxicity. And as you can see, we notice at least that the CAR-28 mediated higher killing capacity for the target here in red as compared to the CAR-41BB in this case, when we culture these CAR T cells at different effector to target ratios over the antigen positive cells. Of course, we characterized these CAR T cells in terms of T cell exhaustion. We mediated higher levels of LAG3 expression in case we used CAR-28 as compared to CAR-41BB. And of course, CAR-28 were able to differentiate to effector cells better compared to the CAR-41BB in terms of the distribution of the T cell subsets among antigen sensitization. To make the story short, we moved one step ahead and tried to look into the in vivo potency of these CAR T cells. And what we did in the beginning is that we established the tumor cells we established the tumor subcutaneously in the SCID mice here in this case, and then we waited until the tumor volume was around 150 to 200 millimeter cube, and then we applied our CAR T cells IV. This scenario did not mediate any tumor control for this mice that injected with the tumor cells. However, what we came up is that this scheme, which is also clinically applicable as well, we have used a combination of chemotherapy called dexataxel, a low dose, non-ablative, and in combination with our CAR T cells. And what we did is that we again established the tumor cells and then wait until the tumor volume around 200 millimeter cube. And then we applied 2 doses of chemotherapy DTX here, IP, and then we waited 5 days and then applied our CAR T cells again IP. To make the story short, we have controlled the tumor very well upon intravenous injection and compared and controlled the tumor better in terms of tumor volume here, as you can see, well, with the DTX plus CAR-28 as compared to DTX and CAR-41BB or DTX alone in this case. So we moved one step ahead and addressed the scenario if we would apply these CAR T cells intratumoral, meaning that a focal application of these CAR T cells and how they behave in this regard. So again, we established the tumor, again subcutaneous in this case, until the tumor reached around 70 millimeter cube or 100 millimeter cube, and then we applied the CAR T cells directly intratumoral. And what we observed is complete elimination of the tumor. Here is a representative mice, and here we look for luciferase signal as indication for the tumor growth in this case. And as you can see here, the untreated T cells injected into this mice did not lead to any tumor control, but rather the tumor spread here in the mice and concentrated more and more. This is the control, which is a mice did not receive any CAR T cells. And here you can see the mice receiving CAR-41BB, where we have subjective responses, meaning that some mice, some tumors were eliminated in the mice, others were under control. However, when we applied CAR-28 T cells, we were able to eliminate all the tumors here, and we did not see any tumor reoccurrence. And this is here the number of animals we applied for each group, and this is the quantification that confirmed that CAR-28 injection intratumorally led to complete elimination of these CAR T cells. So in summary too, we established here a novel CAR T cell platform to target the PSMA, and we have mediated a distinct feature between CAR-28 and CAR-41BB. It is in vitro and in vivo. IV injection led to tumor control in combination with the chemotherapy, DTX, and IT, where led IT injection of these tumors with the CAR-28 led to complete tumor remission. At the end, I would like to thank the people involved in these studies and my colleagues at the Institute, of course, the head of our Institute, Tony Katuman, and Rio for this nice invitation. And of course, I'm happy to take some questions that you might have either now, Andrea, or of course, happy and open to take questions later on from your colleagues or yourself through my email contact. Thank you very much.
Thank you very much, Jamal, for your amazing lecture. I think we already have the first a couple of questions for you.
Yeah, sure.
Thank you. The first question for today is mainly in your opinion, in terms of safety of vectors used in CAR-T cells in a clinical environment. What do you consider is better, the viral, or in the future, maybe the non-viral vectors for clinical use?
Right. Thank you for this important question. I think it's very, very important. And nowadays we have seen actually a couple of cases where the lentiviral integration was seen in T cells and led, for example, to a tumor expansion, and in this case, to leukemic events in the patient. In one hand, for example, it was mediated by targeting, by integration of the lenti CD19 into the TET2 locus. This is at least the published case where it was beneficial for the patient. However, we have seen also that using, for example, sleeping beauty, which is non-viral vectors, led to some leukemia, lymphoma. And the reason is still not clear why this was the case, but I think we have, I believe that we have to be going with a targeted integration strategy. However, this has to be carefully addressed in terms of non-viral transfer, at least in terms of the safety of the nuclease of use. And this is going side by side with the efficacy, of course, and the knock-in efficiency, of course, and how we could, let's say, have a manufacturing of these CAR-T cells without a side effect.
Okay, thank you. So another question is regarding the off-target effects. How do you monitor off-target in vivo, right? In the patient when you are working in the clinical environment.
Right. I think it's rather challenging. This is also again very important question. Thank you very much for this. I guess it's rather here in this case we need an alternative methods to evaluate the safety profile of the nuclease of choice, either in vitro or later on, of course, in the clinically applicable products. And I didn't show you here, but our lab developed a novel and sensitive method, unbiased, to look for all potential translocation happening between the on-target and the off-target sites, and of course the on-target aberration as well. The method called CAR-T, maybe you are familiar with it. Yeah. And this method published recently, and we were able to look for different events of translocation happening between the on-target and the off-target, and of course on the on-target itself. And this method, I think, is very nice because it's highly sensitive, and the need for small amounts of DNA input can be used to mediate and monitor the patient later on, infused with a nuclease treated product in general.
Okay, very good. And the last one is regarding the edited CAR-T cells. Have you worked with just leukemia models or also with solid tumors in edited CAR-T cells?
Yes, I think that the project I showed you for the edited CAR-T cells was for CD19 targeting. So that was for hematological malignancies. However, as nowadays we also have some targeted integration strategy to look for our developed PSMA CARs to see the efficacy and the safety of these product by knocking in the CAR into the track locus, targeting solid tumor, in this case of prostate cancer. And this not yet mature to be published, but hopefully soon will be concluded once we validate the functionality in the preclinical mouse model of these knock-in CAR as compared to the gamma or to the state-of-the-art CAR.
Okay, very, very nice. Thanks again, Jamal, for your amazing and beautiful, very interesting seminar. Thank you for sharing with us your view about the challenge and opportunities in that amazing field. And well, thanks everybody for joining us, and see you the next lecture. Have a nice day. Thank you and bye-bye.
Thank you very much. Bye-bye.